for them to learn, understand, and work with researchers to create new solutions in the dental space. As we've understood from our earlier session in the parallel track, where Uchi was hosting it, that there are a lot of inputs which has come as a feedback from the audience, from students and professionals, that yes, research is the key, and I think that's the key forward in terms of getting these inputs. I'm sorry for the delay, and there was slight technical hitch, but Dr. Santanu is here, and I'm sure it will be a great learning. And you can also post your questions in the discuss panel for you to get more insights and a response from him, so that your doubts are clear. Thank you so much, and may I now ask Dr. Santanu Dhara to please take us through his presentation. Thank you.
Yeah, little but I can do. I could listen. There's, yeah. So thank you for kind invitation, and I think we tried our best to connect. There's a lot of technical glitch, and I think the issues related to network always will be there. I think good afternoon, everyone. So title of my talk is Design, Development, and Validation of Customized Implants Using Ethically Sourced Materials. So I'm using ethically sourced because I'm using the materials which are usually thrown from the usage, or other way the bio waste materials, or sometimes we are using the materials from the earth crust. Mainly we are not harming anyone or damaging anyone to get the materials. And also we are will be talking about customized implant fabrication, right? So incidentally, after joining at School of Medical Science and Technology, we formed the group Biomaterials and Tissue Engineering back in 2007. And our group has 4 major objectives. So digitization of the medical problem, customizing the manufacturing solution, and hunting for the ethically sourced materials. And we do validation in vitro and in vivo preclinical studies for the medical applications for betterment of the healthcare. So for customized 3D manufacturing, so what first thing we do is that, as I mentioned, that digitization of the medical problem, we actually get in touch with the doctor to get the medical data like CT or MRI images for the defects, injuries, or the problems, and then we actually collect the CT or MRI data, and we actually utilize the platform, reverse engineering platform like Simpleware or Thematics or such platform, and from using that platform we extract the implants or the devices or the tissues or the organs exact size and shape. Here I can see a stent, or I can see kind of cartilage in the nose or in the bone with a cancellous bone, similarly in the spine, spinal ribs, and so on. So based on the grayscale value or Hounsfield unit in case of the bone, we extract the images of exact size and shape, and utilizing that image we go for fabrication. Here you can see the couple of devices like dental crowns, dental roots, part of the mandibles, and different fossa and scaffolds. So these are the materials or the design which we have utilized or explored for fabrication of the devices. So we, for manufacturing, customized manufacturing solution, we explore different processes. We broadly go for bottom-up approaches and top-down approaches. In the bottom-up approach, we use direct casting, 3D printing, laser patterning, spinning, electrospinning and wet spinning, and also we go for freeze drying. And in case of rapid tooling, we go for near net shape forming by machining, lamination, machining, etc. And so we actually utilize the combination of the processes to address the challenges, especially for structural and functional aspects of the materials for healthcare applications. So in these aspects, we develop a couple of technologies, platform technologies. By utilizing those, we can make series of devices. We have demonstrated a couple of devices. We will also go into that. So one of the process we developed is protein coagulation casting, mainly for ceramics and metals. Also, we have developed a coagulation assisted foaming for reactive metal foam. Plastic dough processing for dense and porous ceramics. Again, it's a reactive metals mainly. Near net shape forming by green machining. So as you can see here, this is just opposite to 3D printing. In 3D printing, we actually deposit layer by layer and we make the 3D objects. But here what we have done is actually this is a cost-effective solution to 3D printing. Here we have taken the plastic dough processed ceramics and then we machined either side and laminated them to get the scaffolds. Similarly, we have done the CNC machining and then we cast the customized mandible based on the image. As you see here, this is a mandible is developed in our lab. This is a customized mandible, which is patient-specific mandible, and this mandible was made of resin, biocompatible resin, along with ceramic fibers, which is flexible and kind of having property quite similar to our native mandibles. And we also have demonstrated the technology where we can make the customized dental crown by using a mold. So we can machine the mold and where you can cast the composite and you can get the mold and we can get the crowns. Similarly, we developed a process for machining of ceramics and where we have developed the tools, diamond embedded tools, and we developed a green compacts which are machinable. And I think IIT Kharagpur and DBT has a joint patent on that. So we now can make any size and shape of the dental crown, as you can see here, and which is currently I think most of the dental crowns are fabricated, customized crowns are fabricated by following the process developed by Filser et al. And that is actually they use the pre-ceramics, pre-sintered ceramics, and then they machine into shapes. So here what we have done is we take the ceramics and we do the green state machining. And of course we calculate the shrinkages and accordingly we develop the size and shape of the crown. After firing, it becomes very close to the similar one. And then veneering is done and the crown is ready for implantation. Similarly, we have used different biological resources, waste materials as I mentioned, like prawn shell, egg shell, sea coral, placenta, fish scale, and different biopolymers, molasses, green chili, onion peels, and whey proteins for diverse healthcare applications. So just to mention to you, as you see here, fish scale. So utilizing fish scale, we developed series of products and the collagen is one of them. So using collagen, we have made printable bioink, We have made collagen sponge, collagen flake, and now we have a startup, Omnivore Medicare Private Limited, where we are working with a big company for next stage of commercialization for fiscal based different wound dressing products, and we got very remarkable results on that. And I'm coming to a next problem. So implant failure analysis revealed that implant failure mostly happens at the interface between the native tissues and the implant. So as we see here in the image, so like we have an old wall and new wall. If we have a smooth interface, the load transfer is not very smooth. This wall does not interact with the old wall. So there is an effort to put some kind of plates, but it doesn't work. Similarly, if we have the new walls which can cross-talk with the old wall, so then only you can have a nice bonding stitching, right? So in this consequence, in this context, Halvard, a scientist from US University, proposed that if you can use absolutely inert ceramics and if you can make it macro porous, like porosity is 100 to 150 micron, then we can actually avoid the interfacial failure in between the implants and the tissues. So his thought was that because of macro porosity, tissue can grow into the pores and tissue can bind to the implants. So therefore, the load transfer from implants to the tissues or tissues to the implants would be very smooth. So this is what he proposed, the tissue ingrowth, and therefore interfacial bonding to reduce the failure. But Larry Hens, another scientist from University of Florida, he proposed that if we use the bioactive materials, then material can be bound to the tissues, and therefore tissue and material will have very strong bonding. But if you actually look at over the years, so these both the theories was proposed in 1960s, and if you look at in last decade, all the implants came off actually combination of these two approaches. So Halvard says that has to be highly interconnected porous structure, and Haynes said that it has to be bioactive. So if we actually take bioactive materials and highly interconnected one, tissue not only can grow into the pores but also can adhere to the materials. So that could be the approach for the next generation to minimize the stress shielding effect, to offer the biological fixation and better tissue and implant crosstalk. So as you see here, there is dental root which was launched by GMR in 2014. As you see here in the dental root, there is a strategic location where there is an open porous structure. This is a trabecular metal, and here tissue can grow into these pores. Therefore, you have better interfacial bonding. Similarly, you can see here in the mandible. In the mandible, so there is a hydroxyapatite coating. This mandible was implanted in a Brazilian lady. She recovered in 87 years old Brazilian lady, and she recovered in 7 days. So this is because a hydroxyapatite coating, which is actually giving the bioactive surface, therefore much faster bone bonding. Similarly, you see the series of hip joints. So you can see the hip implants. So earlier hip implants was very smooth, but now scientists has realized that actually it has to be rough at strategic locations. So in fact, the implant which is interfacing the tissues has to be rough and macroporous. Zimmer has given the term osteoincorporation because tissue or bone can grow on the surface of this porous matrix, as well as bone can grow into the pores. So on growth and ingrowth combinedly called osteoincorporation, which I have mentioned in the previous slide, biological fixation. Then what will happen is bigger volume of materials can crosstalk with bigger volume of tissues. Therefore, the stress yielding can be minimized and load transfer would be much smooth. So in this context, we developed a couple of technologies. One of the technology is a plastic dough processing, where we have developed combinatorial approach of fabrication and surface modification, by which we are able to improve the interfacial bonding, and we are able to make series of products like we can see the hollow tubes and the kind of logo dense cylinders, uh, dental roots, and so on. But usually, otherwise, very difficult to make these objects by conventional route of machining, as well as so titanium processing itself is you have to use very high vacuum and very sophisticated instruments. But we have proposed here a very simplified method by which we can make the titanium. So as you see here, we have granted US patent and Indian patent, and we also developed a porous titanium, and we have implanted in animal. You can see that tissue can grow into. But this porous titanium is having randomly aligned porosity. By 3D printing, we can actually make the porous structure as per our need. So we can program them. We can make as per our design. But however, actually biological tissues actually favors the random structure. So that way, so we have made the random structure, and also we have made the 3D printed structure. So we have done with the tissues, and this is a process which is powder metallurgy based process. We have done series of products, and as you can see from microcity results, so we are able to make porous samples which porosity can be varied from 45% to 90%. So we have made series of spinal spacers, and this spinal spacers design was borrowed from Zimmer, and this project was done under the Bioactive Srishti sponsor project. And we have shown that using this porous titanium, we can make the spinal spacer. And currently these porous titanium spacer are actually marketed by Zimmer, and there are some other companies. But in India, there is no such indigenous company who is making actually porous titanium. So this porous titanium we have actually implanted in animal, and we have shown that how with the varying the porosity, the bone ingrowth can be varied. As you can show here, with the higher porosity, we can see that with the 92% porosity, we can see that bone volume inside the porous substrate. I think this is 92 and 190. 178 and 297 are the pore sizes. So as I mentioned here, that pore size is very important. Pore size has to be, as proposed by Halbert, 150 to 200 micron. Actually, this number is very critical because when tissue has to grow into the pores, vasculature has to form. For forming the vasculature, minimum pore size requirement is 100 micron. But we have shown that if we play with 92 micron, 178 micron, 297 micron, 297 micron pore size has much better bone volume inside the porous titanium as compared to S92. So then we have also, I think this we have evaluated with microcity as well as with histological studies, and we have seen the remarkable result. Now I'm coming to the 3D printing of titanium. So we have used a titanium-based slurry, and then it was printed using a printer. This printer was developed by us in the laboratory, and utilizing this printer, as you can see here, we made the 3D lattice structure. Here also we played with the lattice parameters, and we have seen that tentatively around 600 micron lattice or strand gap that helps the maximum tissue ingrowth. So we have done in animal, and as you can see here, the location where grafted implant has a very nice interaction with the bone, there you see significant bone ingrowth into the implants. Wherever in the other side, because there is a gap between the bone and the implant, so there is no significant bone ingrowth. So this is shown that on the start there is a huge cell sheath, cell growth on the surface, and as you see from the 3D printed structure microcity result, that there is a bone ingrowth, and as you see there is also vasculature. So therefore we can say that this 3D printed structure is good for um, kind of tissue ingrowth and fixation. So of course this can be tried with the devices. So now we are exploring those part. So another interesting idea is egg cell and sea cell, which are actually biological waste. And as you know, in the Mumbai coast or maybe in our IIT Kharagpur 100 kilometer away, there is a coast where there are huge amount of sea cell is there. And also we can see the egg cell which is there is a biological waste. I think there are couple of papers or plenty of papers where they are exploring the hydroxyapatite synthesis utilizing this kind of egg cell and sea cell. But here we are using a different approach. Instead of utilizing this as a raw material for powder synthesis, what we have done is we have taken this powder, mixed with phosphoric acid, made a ink, printable ink, and then we printed into the scaffold. And then as you fire, you get the direct device. So you can step jump and you can make the device. So as you see here, we made the 3D printed structure and then directly we printed and we got the scaffold. And this scaffold was compared with the chemically derived hydroxyapatite scaffold, and we got much remarkable result with Excel based samples as compared to the um, chemically derived one. So in fact, we got in C cell and X cell both, we got better result than the chemically derived one. Perhaps it is because the chemically derived one has a perfect hydroxyapatite structure. However, in case of C cell and X cell based materials, there is a stoichiometric defects that may shows the better reversibility of hydroxyapatite, which is actually in vivo degrading much faster, giving us higher concentration of calcium and phosphate at the site. Therefore, it is promoting us to deposit better and deposit the bone-like structure on the site much better. So we have done with the animal, and of course we have seen that you can see here the big gap which can be closed using the 3D printed scaffolds. We have used the placenta, for example, and using placenta we have decellularized and we have used cell-free and cell-based gel which is placed in the cartilage, and we have seen that in case of cell-based placenta matrix we got much faster cartilage healing as compared to the empty defect and cell-free matrix. Similarly, in the wound dressing, we have a lot of activities. As you know, the skin is a bilayered structure where there is a connective tissues and or maybe multi-layered structure. There is a fibroblast, connective tissues, and there is a epidermal layer. And in between, there is a basement membrane. So if you can make a device where fibroblast and keratinocyte, they are compartmentalized, yet they can interact. And this in-between layer can be a antimicrobial, and it can inhibit the bacterial invasion and microbial invasion. So then you can make a better device. So with this idea in mind, what we have done, we have used electrospinning as a method, using electrospun membrane, and we have infiltrated the collagen. And so on the top of the electrospun membrane, we have grown the keratinocyte, and on the bottom side, we have grown the fibroblast. And we have done a testing with a skin equivalent model. As you see here, without presence of collagen, there is no keratinocyte migration, but with collagen, there is a migration of keratinocyte. So it shows that if you have collagen with a bilayer structure, it is giving us better recovery of wounds as compared to the without presence of collagen. Similarly, we have taken another kind of idea where we do the electrospinning to make the nanofibers, but if we make the emulsion, and this emulsion can be used for controlling the pore size. For example, if we make the emulsion, if we mix for 12 hours and then you do the electrospinning, we get the membrane. And if you mix for 5 minutes, and then if you do electrospinning, we get floppy mass, because here the droplet size is much bigger, and that is giving us floppy mass. Now, on the top of the floppy mass, if you can have a kind of nanofibrous membrane, then you can have a controlled porosity. And then if you can infiltrate collagen in the macroporous site, then you can actually have a bilayer structure. And this can be used as a skin graft. As you see here, we have done the study and we have done the co-culture. You can see there is a compartmentalization of keratinocyte and fibroblast, and we have done some kind of gene expression and transcriptomic studies, and from there we find that ecadherin, laminin, collagen 4, involucrin, and keratin 14 and keratin 10 expression. It shows that this works wonderfully, and also there is a significant amount of skin maturation. And this kind of skin graft we have tested with animals, and we have seen that in 21 days time there is a complete healing of the wounds. Therefore, this kind of matrix can be utilized for wound healing purposes. And while we have done the immunohistological studies, we have seen that we can have a significant reduction in scar utilizing the collagen. And usually this collagen we are using from fish scale derived collagen. And of course, we have used different biopolymers like chitosan from crab cell or shrimp cell. And these chitosan nanofibers were fabricated by modification with the allyl chitosan. And these kind of polymers, simply because of antiphilicity, you can do phase separation and you get the nanofibers. And these nanofibers were mixed with the carbon dots having fluorescent properties. And when this carbon dot based loaded nanofibers are grafted on the skin side, right? And you go for imaging application, so we can see the fate of the wounds at different time points without touching the wound bed. So therefore, non-invasive way we can monitor the wound, and we know that these nanofibers as well as these carbon dots has the wound healing efficacy. And these carbon dots were explored for live cell imaging. So we could see that we can track these cells using these carbon dots, onion peel based carbon dots, and green chili derived carbon dots, whey protein derived carbon dots. And these carbon dots were able to track the cells up to 8 generations. However, it doesn't have any bleaching effect. It is not toxic apparently. Okay, so therefore this can be wonderfully used for this kind of applications. And of course, there is another interesting work which we have done, that as you know that sometimes by mistake some I think for the surgery doctors are using the radiopaque tracer, so that we can trace if anything by mistake it is inside the body, something left or left over, like gauze or something else. So there are some radio opaque tracers. So what we have done, we developed the radio opaque polymers, and that polymers can be used for diverse application in therapeutics. So like for example, if you take a catgut suture, and this catgut suture usually is a catgut suture is nothing but cat intestine which is cross-linked to the chromic acid, and this has a possibility of infection because of disease transmission, because of kind of um, um, infection. So and so this catgut simply can be dipped in this kind of DHF, a butyl kind of compound in presence of iodine mixed together. And when you dip in, so what will happen? They will not only cross-link in situ, but also they will iodine. and as you see here, the GI tract can be imaged using this kind of radio opaque beads. And also use the goat ear at a collagen 2. Not only that, we have done some kind of gene sequencing, amino acid sequencing. From that, we can predict early there is any kind of if there is any kind of disease transmission, if there is any kind of symptoms of arthritis. Okay, so we can predict that if there is a kind of inflammation towards arthritis, osteoarthritis, that can be predicted. Okay. So here what we have done, we have extensively utilized this proteomics, multi-top platform, and then we explored different peptides, and from the sequence in the peptides, we are able to predict, or we are able to identify the early markers for any initiation of osteoarthritis. Okay, and of course we have also developed an injectable gel, where injectable gel and cells combinedly we can inject into the joints where cartilage can be revived. So we are still working on this. Okay, in nutshell, we have developed a couple of technologies where we are able to make the customized dental crown. We are able to make the 3D printed scaffolds utilizing egg cell, C cell, as well as titanium. And we have made the customized mandible. We have developed platform technologies for porous and dense titanium, as well as porous and dense ceramics. And we have also developed carbon dots, which can have a therapeutic value as well as for imaging, can be utilized. And we have used placenta for wound healing as well as placenta for cartilage healing. And we developed that radiopaque polymers can be used for imaging purposes. So I would like to thank all the organizers as well as the people who are listening to me. And I'm thankful to DST, DBT, MHRD, DRDO, IIT Kharagpur for the supports, and BIRAC for the funding. So if you have any queries, please let me know. Hello. So any queries?